Okay. Hello and good evening, everyone. Welcome to the fourth plenary of SCCS-Bengaluru 2021. Our speaker today is Professor David Borchers, a statistician from St. Andrews University. He's going to be talking to us about ecological inferences from sparse data. To introduce Professor Borchers, I would like to invite Dr. Kaustubh Sharma, Assistant Director at Conservation Policy and Partnerships at Snow Leopard Trust, to share a few words about Professor Borchers. Kaustubh?
Thank you, Nupur, and good evening, everyone. Thank you, organizers at SCCS, for inviting me to welcome the Professor David Borchers. As I mentioned a few minutes ago, when we were having a chat with Nupur, SCCS has evolved into an institution today, and I'd really like to congratulate them for not only providing a great platform to students to connect, share, and learn, but also to engage with experts in diverse fields. Professor Borchers is one of the kindest, gentlest, yet the sincerest advisors one can get to work with. I was fortunate to meet David in 2016 in Seattle, where I was presenting at the International Conference on Statistical Ecology. David was already a celebrity there. He was already busy chairing, mentoring, and questioning when his colleagues, one of his colleagues, an old friend, Dr. Len Thomas, introduced me to him as David was kind of trying to take a break away from the bustle. Basically, it was Len getting me off his back for bugging him about some half-baked ideas about spatial capture, recapture, and snow leopard abundance. Now, David spent time hearing me patiently, as he always does, and asked me to first learn more about the methods. He invited me to attend one of his courses on spatial capture, recapture, before even trying to jump around with those ideas and asking for help. I think that to me was David teaching me how to fish rather than handing me over the fish. If anything, it was the beginning of an exciting collaboration that is still going strong. As you all know, Professor David Borchers is the director of the Center for Research into Ecological and Environmental Modeling at the University of St. Andrews. His research has focused on developing statistical methods for estimating wildlife population parameters using distance sampling, mark recapture and removal methods, sampling theory, and spatial modeling methods. David also chairs the technical advisory panel of a very ambitious initiative, which we call as the Population Assessment of the World's Snow Leopards, in short, PAWS. And in the last 5 years, David's supervision has led to the snow leopard conservation fraternity on a path that was earlier presumed to be impossible. Now, just imagine who could have thought of surveying snow leopards to represent 2 million square kilometers, bringing together 12 sovereign countries, several NGOs and universities on board with a unified methodological process. Today, David is also known as the snow leopard man among the statistical ecologists. He's already a pro at winning cakes and beer at quizzes that include snow leopards and other big cats, and has been beating even ecologists at the game. David is not only a sound and accomplished statistician with more than 105 peer-reviewed publications, but he's also an outstanding teacher and communicator. Albert Einstein once said, if you can't explain it simply, you don't understand it well enough. The ease with which David explains the nuances of statistical ecology to a diverse audience is a treat to watch, hear, and read. From conservationists to government officials, from students to ministers, people get hooked to the magic of statistical ecology whenever Professor Borchers is talking. And like every other time, I cannot wait to hear David speak today and learn a thing or two new. And yeah, with that, I'd like to just shut up now and hand over the mic to Professor David Borchers, the daddy of spatial capture recapture. Over to you, David.
Thanks very much, Kaustubh. And I'd like to first start by thanking the organizers for inviting me to this. It's a real pleasure to be here, and I really have looked forward to it and continue to do so. Then I have to respond to some of the things that Kaustubh said, because those of you who know Kaustubh know that he is unreasonably generous with his praise and assessment of his colleagues. So I'm not going to live up to really fantastic billing that he's given me, but thanks very much anyway, Kostya. So with that, I think I'll just move right on. I'll share my screen. And I think you should now be able to see.
Yes.
The screen? Okay, so let me go ahead then. So I added one word to my title. I added spatial, because when I came to write the talk, I realized that everything I was talking about was really relating to things that are spatial. I also need to start with a bit of an apology in a way, in that my knowledge of ecology is very limited. My knowledge of conservation is even more limited. So I have no expertise in those areas at all. As Kostya said, I'm a statistician, and it's by collaborating with people like Kostya and other ecologists and conservationists that I'm able to have any impact whatsoever on these important topics. The logos down the bottom here are organizations that have contributed to some of the work that I'm going to talk about. I will, as I go through examples, I'll acknowledge individuals and organizations that contributed to the work. Okay, so moving right in, what struck me when I was thinking about this talk was how sparse the data is from which we try to make inferences about ecological wildlife populations. And the next thing I thought, well, technology is changing things so much with human populations. It would be interesting to contrast what we know about human populations to what we know about animal populations and the different kinds of data we get from the two. So I managed to find this little video over here, which shows the Trump supporters storming the US Capitol. The little green dots are mobile telephones, so we know pretty much where every single one of these supporters were. But I want you to think of this, I'll play the video in a second. I want you to think of it as an ecological population. So it's a population that lives in a heterogeneous environment. There are paths that are easy to go down. There are obstacles in the way. And the things that I'm going to be talking about in my talk, which I want you to think about as I play this video, is think about the distribution and habitat preferences of this population of Trump supporters. Think about their movement, how they move about, and the connectivity, so where they move to get from A to B. Think about the change in abundance that happens from the beginning of the video to the end of the video. So the population declines towards the end of the video, as you will see. And finally, think about the activity pattern. So when are this ecological population, when is it active and when is it inactive? Okay, so I'll play the video. So they start off with a very big habitat preference. We are quite inactive, and then they're going to move off, and you can see they have certain preferences of ways of moving, connectivity. They spend some time here, and then the population declines. They will go home. They leave this, if this was our ecological survey area. So there we can see pretty much every individual, so we know what the population is doing. Now contrast that to the sort of thing we get with ecological surveys. And I've ended up in this talk talking really only about spatial capture-recapture methods, just because that's what I'm doing most of at the moment. But the messages I think carry over to any kind of ecological survey you do. So this is a spatial capture-recapture survey of some jaguars in Belize. These dots are the cameras, and when the dots have a color on them, that's an animal that was detected. So this animal over here that I'm going to call animal number I was detected there, 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 and there. So if we think of this as the Trump supporter population, then there's the heterogeneous environment shown by this habitat index in the background. And this individual in the population is individually recognizable. So we had someone sitting in a building here, say a building there, building there, building there. And each of these people saw this individual pass. So they saw them at these 4 times. And we have that data for a few individuals. You can see there are not many colors here. We don't have it for the whole population. We don't have it all the time. And for all the population out here, we really just don't know what they're doing. So we have just much, much less data, far more sparse data, but we want to draw inferences about the whole population. So a central question in statistical ecology is how do you find out about the things that you didn't detect from the few that you did detect? And the answer to that question is you assume that both were generated by the same underlying process at the population level. And this underlying process has randomness to it. It's a statistical process. So before I get into the underlying process, I just want to go through a simple survey situation. If you've ever done a course in sampling theory or sampling statistics, you will have done this kind of thing. We're going to survey this area. There's some habitat covariate there we're not going to talk about for the minute. We put down a square, and let's just say that's 1 kilometer square. And we see some things in that square. And what links the things in that square to the things everywhere else is that we randomly place the square so that all animals, whether they're detected or not, had the same probability of being detected. That's what they share. We can estimate density. If we saw 10 here, we estimate there were a density of 10 per square kilometer. We get no measure of uncertainty. So if we do some replication, we survey 2 other areas, we see 6 and 14, our density estimate is still 10. And now we can estimate the uncertainty associated with that just by this usual formula for estimating variance of a number. So no statistical model yet, we're just estimating a density and a variance. In order to make probability statements or get confidence intervals, we need a model. And usually for this sort of thing, we would just say, well, our density estimate may be normally distributed, and we rely on some large sample theoretical results to hope that that's true. So this is not the sort of survey I want to talk about. All the randomness in this survey is from the design. It's where we placed the squares. The design links the things we saw to the things we didn't see. You can show theoretically that you get unbiased estimates, so that's good. But because there's no model for the underlying ecological process, we can't draw any inferences about the underlying processes. We can't say anything about distribution, about movement, about habitat use, or any such things which are of interest to ecologists and conservationists. So we don't do that kind of survey. What we do is this. We do what's called model-based inference, whereas what I've just described is called design-based inference. And the key to that is this expression here. We come up with an expression for the probability of observing the data that we observed, whatever that was, given the underlying process that we hypothesize. And this has some unknown parameters in. Once we've come up with this model, and that's what us statisticians do, we specialize in developing exactly this sort of probability statement, we can then estimate things about the underlying process. And there are 2 broad ways of doing that. One is by maximum likelihood, where you just find the most likely underlying process among those you're considering to have generated the data you observed. Or if you're Bayesian, you update your beliefs on the underlying process in the light of the data you observed. Okay, so what do I mean by underlying process? Well, I'm going to give some details, but I'll just start off with a very simple example. It usually has 2 parts, almost invariably with ecological surveys. It's got the ecological process, so we have some statistical description of what's going on ecologically. And in the very simplest case, this is just animals have a density D. And then we can write down some statistical model that involves D. So the number of animals depends on D through some Poisson process, and their locations depend on, or uniform in space, for example. I'll be showing very few equations. They're not central at all. I just wanted to show that D appears in this. So we have some parameters that relate to the ecological process, but then we also have parameters that relate to the detection process. For example, the simplest would be all animals are equally detectable with some probability P, and we can write down a distribution for that. And this parameter P is called nuisance parameters, or parameters when there's more than one by statisticians, because they're a nuisance. We're interested in D. We usually aren't interested in P, but we have to estimate P in order to get to D. So it's a nuisance. We'd rather not deal with it. But it's a special kind of nuisance. And I attended a talk a while ago by Todd Arnold of the University of Minnesota, and he said, these are not nuisances in the way that flies hovering around your food are nuisances. These are nuisances like zombies, and poisonous snakes are nuisances. Ignore them at your peril. So these nuisance parameters we're going to have to pay attention to, and they actually turn out to be quite informative in some instances, which I'll describe in a while. Okay, so I'm going to go through some case studies now illustrating how these statistical models tell us ecological things. And I'm going to start with the simplest sort of case in which we're just going to make density depend on some habitat covariate. So this is a camera trap survey done by Nakedi Maputla for his PhD in the Kruger National Park of leopards. And we're just going to be looking at this little bottom section of the Kruger National Park, which is shown here again. The pins are camera traps. The coloring is a habitat covariate that was constructed on the basis of vegetation types. So ecologists ranked the vegetation types in terms of how suitable they thought they were for leopards, and then we just smoothed through their ranking, and that's the coloring that's shown here. And then we fitted a model in which we let the density depend on this habitat covariate, which I'm calling Z. So this is a log linear relationship between the habitat covariate and Z. And we assume some distribution for the observed data. That's not what I want to talk about. We estimate some parameters. And in particular, we estimate this beta-1 parameter, which is the effect of the habitat covariate. So if this is a positive number, that says that the habitat is positively associated with density. The higher the covariate, the higher the density. And the picture on the left here shows the height of these bars is the estimated density, and the color is the habitat covariate. So you can see we've now got some ecological information about this population. They like living in this kind of habitat. They don't like living in this blue kind of habitat so much. So that's a pretty simple model, but we can get it more complicated than that. So here's another spatial capture survey, this time with hair snares and genetic identification. And it's work done by Lamb and colleagues and referenced in this paper at the bottom here. So this is the survey region shown here, and the survey region is shown again here with the color on the base being the habitat preference covariate, red dots being the hair snares, and the height of these pins being how many animals were detected there. So you can see there's definite non-uniformity in the population here. But out of just these data, plus the nuisance parameter data, we want to estimate what sort of things, what sort of habitats bears like and what they dislike. And the covariates we have are this habitat covariate, which is shown on the left here, a road density, which has been stratified into two classes, low road density in green, high in blue, and whether or not you're in a national park or not. So parks are green, gray is not parks. So those are our three explanatory variables. And we can fit very much the same sort of model that we fitted to the Kruger Park data. And for the moment, just focus on this equation here and this picture on the right. They go together. So here we've modeled density, just like with the Kruger Park, as a function of habitat, but also as a function of this road density covariate. And it's a log linear function, so it's shown here in gray. What I've got is on the x-axis is the habitat suitability index. The ticks are where the hair snares were. And this beta-1 parameter is positive, so it says as habitat suitability increases, so does density, and it increases in an exponential way. And that exponential increase leads to this really high density spot here, because the habitat covariate is high there, it's good, and it's also low road density. But that turns out to be too simple a model. So we fitted another model in which we allow the density to have a turning point. So it increases as habitat suitability increases, but then it can turn over and decrease. And when we fit both of these models, so the second model is described here, and the thing about it, you will notice, forget the form that's shown here, the thing to notice is there's one more parameter here than there is there. So by allowing a little more complicated model, we can estimate a different kind of relationship, and then we can use sort of standard model selection techniques to decide whether this model is better than that one, and it turns out that this model is in fact better. So the habitat suitability index, they didn't get quite right. It makes density increase up to a point, but then beyond that point, it tends to decrease. It also turned out that this road density covariate was important, so you can see its effect here, but it turned out that the park effect wasn't important. So not only does this give you a model that explains to some extent why animals are in certain places and says that they don't like high road density, it also could be used to test hypotheses like, is the national park, are the parks increasing the animal density or not? Okay, now in that example, we actually also fitted some models to these nuisance parameters. Remember the nuisance parameters? We're interested in density, but the nuisance parameters we have to estimate in order to estimate density. And the two nuisance parameters here are to do with the encounter rates. So this lambda 0 thing is the expected number of times you encounter a bear if you put a hair snare at its activity center, so where it spends most time. The sigma is an index of how far the bears roam from the activity center. And we fitted a variety of models with increasing bendiness as you go from the left to the right here. And the models that were selected by model selection criteria are these two. So the expected encounter rate at the activity center starts high. Or I should say there are actually 4 occasions here. So it goes 1, 2, 3, 4 occasions. They're separated by about 2 weeks. So on the first occasion, the expected number of encounters at the activity center is high, drops low to the middle 2 occasions, then goes high again for the last one. And the range parameter, how far the bear's range, does the opposite. It starts low, so they're not ranging far, then it goes higher for the middle 2 occasions, then it drops to fairly low again on the last occasion. So although we call these nuisance parameters, this actually tells us something quite interesting about what bears are doing. It says that in the circumstances, whatever they were, that applied on the first occasion, the bears tended to spend a lot of time near the activity center, that's why this went up, and didn't range very far from the activity center, so this was small. In the middle 2 occasions, they ranged much further, and as a consequence, they didn't spend as much time at the activity center, which is why this goes down. And then on the last occasion, they again spend a bit more time at the activity center and didn't range as far. Now, I don't have explanatory variables as to why that was, other than occasion here. But with suitable explanatory variables, maybe there was a weather effect, or maybe, I don't know, something else that would be driving this behavior. The point is that we can estimate the behavior from these nuisance parameters and from a relatively simple statistical model. Okay, but this assumes this. If you're an ecologist, the first thing you should worry about this would be the single sigma here, because it says there's only one range parameter. It says bears range a certain distance. It doesn't matter which direction they're going or what habitat they're in, they range that distance. So that seems implausible in a heterogeneous environment. So we can be a bit more sophisticated about that. And this is a particularly nice project to talk about at this conference, because it arose from an SCCS student internship with me at St. Andrews. This was maybe 4 years ago. So Manjari Roy came and spent, I think, 6 weeks or so at St. Andrews, and I worked with her a bit, and she brought this data set along, which is reported in this paper cited at the bottom here. And it's on tigers in Sundarbans, which is an area in this part of India, and zoomed in looks a bit like this. So the features, you can see it's very heterogeneous, and in particular consists of land, the green, and then waterways. And these brown areas are shallow waterways. The blue is deep water, and the tigers do move through the shallow waterways, but they also move over land. So you would expect their movement to depend very much on what their environment looked like in terms of, is it water or is it land? And so we fitted that kind of model. Now, Manjari and colleagues had put out camera traps, which are, you can just about see, are the red crosses here. And they've got circles around them when a tiger was detected. And tigers are linked by lines just the same way as the jaguars were in the second slide I showed. So we have two covariates here. We have a binary covariate, whether it's land or water, green land, blue water, and then also used a covariate distance to water. So here is far from water, and then obviously the blue is right in water. And we're going to fit models in which their movement depends on these covariates. So not where their density is, but where how they move about their home ranges depends on these covariates. And they are known to move through shallow water. And also there's an expectation that males and females have different home range sizes, with females having smaller home range sizes. So those are things we'd like to be able to capture in our model. And the way we're going to do that is we're going to use this idea, which is due to Andy Royal and colleagues, and is in this paper cited at the bottom here, of least cost distance. So I'm going to illustrate the idea using this picture on the left here. So here's some notional simplified region in which I've got something that's called resistance. So the light gray is low resistance, and animals can move through it quite easily. The dark gray is high resistance. They don't like, well, it's more difficult for them to move through. The white, they can't move through. It's an obstacle they can't get into. And we have an animal that starts top right and wants to get bottom left. Now, if you look at the animal's shortest distance, is clearly just a straight line, which is called Euclidean distance from top right to bottom left. But when you take account of the resistance, which is what's done in this bottom left picture, it's actually more efficient for the animal not to take the shortest Euclidean distance, but to go around sticking to the low resistance bits. So if we had an animal who lived here, who wanted to get there, the animal is going to do this because that's notionally the lowest effort as opposed to the shortest distance. And this picture just shows distance in another metric, not least cost. It's another kind of distance. So we're going to scale distances by how much resistance there is along the path. So this distance becomes long because it goes through high resistance. And this distance, one shown here, is short because it goes through low resistance. So we'd expect in the tiger example that there'd be higher resistance to movement in water than on land because we think tigers would prefer to move on land, but they will move through water. And we're going to add a single parameter, this alpha thing. We don't know what this resistance is. All we know is whether it was water or land. We're going to add a parameter alpha that will quantify the resistance. So if alpha is 0, then water and land are equally resistant. If it's positive, then land is less resistant. So we've just got to estimate one more parameter. So when we do that, we get these results for males. So focus on the leftmost picture here. All I've done here is I've said if there's an activity center at the green dot for 3 different arbitrarily selected green dots, and the animal wants to move to the red dot, and the 2 red dots are equidistant, so the same Euclidean distance from the animal, how would the animal go according to our model where we've estimated this resistance parameter? And in this case, the habitat is all land, so the animal will just move straight there, straight left, straight right. In this case, it's got to get from green to red, but there's water all the way in between. If it goes south, there's no water if it goes north. And so the distance and the route it takes going south takes a circuitous route. It keeps more on land. It doesn't keep totally on land because there it crosses a bit of water. So it can move through water, but this is a longer distance to the animal than this one because there's water in the way, and water is resistant to movement. Resistant being the animal doesn't really like moving through it. This one is more extreme because to get from green to red here, it has to go across a large patch of water, and we estimate it's not prepared to do that. So it would take this route. It would go way north, then cross over a little land bridge, then go way south. So although they're equidistant in Euclidean distance, this red dot is much further for the animal than this one. So when we've estimated this parameter, we can then look, well, if the animal lives here, how much time would it spend at various distances from where it lives? And the shading here shows that. So it's the expected number of encounters at any distance away from this, from the activity center. And you can see that blue, even though this blue is right next to where the animal is, blue is far away. To an animal, getting into the middle of this sea here, or the water here, is a distance it's not prepared to go. It is prepared to go through a bit of water because look, there's water here and there's non-blue distance here. So it will go there, but it's very reluctant to. The distance to the animal there is quite big. This one's pretty symmetric because it's pretty much all land. And this one avoids this big chunk of water over here. So we've managed to reproduce fairly complicated movement behavior within the home range, just using this single extra parameter on the same data, really limited data. And not only that, but we can distinguish between males and females. So the effect of a range was different for males and females. And these are the same pictures shown for the females instead of the males. And you can see that their activity center, like their home ranges, are much smaller, but they do the same sort of water avoidance behavior. that the males will do. Now this we did with Kostov, actually, with snow leopards. And I haven't shown the cameras here, but this is a camera trap survey was done in this region. The yellow and browny bits are high ruggedness. The green is low ruggedness. Snow leopards don't like low ruggedness. They like high ruggedness. So after estimating these resistance parameters, I've just made a few of the similar sort of picture. If an animal wants to get from a green dot, so green dot to a red dot, how's it going to get there? And you will see that it is predicted to get there by sticking to rugged areas. It follows this sort of spur of ruggedness rather than taking the shortest distance. To get from here to here, it wouldn't go the straight line distance. It would follow the spur of ruggedness, hop across, more rugged, hop across. So exactly the same sort of behavior. And the nice thing about this particular data set is that we have some GPS tag data that we can verify that this is what the animals are doing. And I'll show you that in the next slide. Just to notice, in the next slide is covering the same region, and the way you can orient yourself in that slide is there's this semicircular high ruggedness area here, and then this large patch of ruggedness over here. So remember those. And this is showing now movement data from tags attached to a snow leopard and a cub, thanks to the organization Slow Leopard Trust and the Conservation Foundation of Mongolia. So here is the semicircle of ruggedness. And here is that high ruggedness patch that we saw in the last video. Oh, not video, the last slide. I will now show you this video though. So this video is the snow leopard mother and cub moving around their habitat. And what we've estimated by using that least cost distance is they're going to stick largely to the rugged areas. They're not going to cross flat areas or be reluctant to. So let's just see what they do. And they do exactly that. They stick to the rugged areas. They do occasionally cross the flat area, but they will tend to go around the flat area. So the behavior that we managed to estimate just from simple camera trap data using this least cost distance is behavior that is verified by GPS tag data, where, like the Trump supporters, we can see exactly where they're going. Okay, moving to a slightly different topic. That was movement within home range. What I'm going to look at now, and over short periods of time, I'm now going to look at a population trajectory of a population of jaguars in Belize over a 12-year period. So Panthera conducted this 12-year camera trapping survey in Belize. The camera locations in 1 year are shown as these dots over here. And this work is reported in the publication shown at the bottom here. So we're interested in now is what's happening to the population and the demographic parameters, birth and death rates. The data, we got 53 males detected on average only twice per year. So this is the picture I showed at the beginning. It's somewhat heterogeneous habitat, but not very. The dark is forest, and it gets less and less foresty. There are also rivers and tracks that the animals use that I haven't shown, but are relevant later. So we have 12 years of that sort of data. And what's our model going to look like? We now need a model that includes births, deaths, and the evolution over time of these things. So we're going to have a very simple population dynamics model. We're going to start with some notional unborn pool of individuals. So these are the potential individuals who either exist but not in the survey region, or don't yet exist and are going to be born. Then we estimate that they're born with some parameter which may depend on year. K is year here. And they go from the unborn pool then into the alive pool. And then they survive in there for some time. And then after some time, they either die or they move out of the area. And they go from the alive pool to here. And when I showed this graphic to a colleague of mine who's pretty much the same age as me, he said, "What's Spider-Man doing there?" So I'll leave you to figure out why Spider-Man is there. But the thing I want to mention is that we have another parameter here. This is the survival rate in year K. So the probability of dying is just 1 minus the probability of surviving. And this is apparent survival because we don't know whether they died or they just left the region. Okay, so we're going to go ahead and fit that data to these 12 years of, fit that model to 12 years of data. And when we do that, we get this blue line shown here, together with the blue shaded area is the 95% confidence bounds. That's fine and well, except that when we fit the same kind of model, same kind of spatial capture recapture model to the individual years, we get this red line. And the striking thing about that is the blue line is pretty much always a good deal below the red line, and that shouldn't be happening. Something's going wrong there. And it turns out that we didn't model the activity center movement adequately. So when we put in a model for diffusion of activity centers, then the fitted to individual year model, the red line, coincides very well with our population dynamics model, the blue line. And note that we've reduced the width of the confidence intervals because we're using all the data over the 12 years to estimate this trajectory, whereas here you're estimating separate abundances every year. So that was a bit of a surprise. We didn't anticipate having to model anything other than birth and death when we did this. And it took us a long time to figure out what was going on. But in the end, we figured out what was going on was activity centers were moving between years. So why does that matter? Well, I'll illustrate that graphically here. So in order to deal with it, we added a model for activity center diffusion, so just randomly drifting. And there's another parameter now that we have to estimate this diffusion. So I'm going to show a very simple-minded little animation here of what is going on. So this is the animal's activity center, and this is its home range in 1 year. I'm just going to show the animation for 6 notional years, and you'll see what happens. So the animal activity center drifts between years. So over the 6 years, it's done that. Now, if we ignore the fact that the activity center has drifted, and we just say it had a single activity center over this whole time, which is what our initial model said, then in order to encompass the movement that it did, we estimate that its range is much bigger than its real range. So its real range is the radius of this blue circles. Our estimated range is the radius of the reddish, pinkish circle. And by having estimated it has greater range, we therefore estimate it is more detectable because it is detected by moving. The further it moves, the more often it's going to be detected. So with the red model, we've overestimated its range, overestimated its detectability, and therefore underestimated the abundance because we think we've sampled a higher proportion of the population than we have because we think they're more detectable than they really are. So here are the estimates from the two best models. The best model, by the way, didn't have a year-specific survival and birth. It just had a single rate. So this is a model in which activity centers are not allowed to move. This is one in which we have a diffusion parameter, which is shown in the bottom here, for them moving. And the most striking feature of this is that the range parameter has gone down by about 60% when we allow the activity centers to move. So allowing the activity centers to move has done 2 things. One, it's let us come up with a much more reliable estimate of density, which has gone up by 25%, and that's why it now agrees with the individual year estimates. But it's also told us something about the ecology of the animal. It's told us that animals' activity centers will drift, even though the animal is territorial and it will live in this region, or it may have lived in this region for 10 years. The activity centers do have a tendency to move between years. And then also by allowing that part of the model, we've got changes in the 2 demographic parameters that we were interested in. So the survival rate has gone down a little bit, and the birth rate, the apparent birth rate, has gone up a little bit. Okay, the other thing we do have from camera trap data, which is very seldom used, but which is informative, is that we know when each detection occurred at each camera trap. So I'm now switching from looking at big picture over 12 years, I'm now switching to very small picture, looking at within a day, how does that animal's activity pattern change? And because the cameras will give me exact times of detections, I have information that'll help me figure that out. So let's look at this ith animal again. So this guy was detected at time TI1, then he went missing for a while, then he appeared there, then appeared there, and then appeared here. Now, although these are straight lines, that's not where the animal went necessarily. These are just connecting, indicating they're the same animal. We know the animal is here, then we don't know what happened to it. It went somewhere, then it appeared there, went somewhere, appeared there. But what we're going to do for the moment is we're going to try and use these data to tell us about diurnal activity patterns of animals. And so when we put in a few extra parameters, so we now allow the number of encounters to depend on time of day. And fitting to these data, we had to use 3 parameters because the relationship between time of day and number of encounters turns out to be quite nonlinear. And it also turns out to be slightly different for males and females. So here's time of day along the x-axis. That's midnight. This is 11 p.m. in the night. This is midday, right here. And yes, these are males' expected number of encounters, which is a direct measurement of expected activity because the encounter occurs because the animal is active and walks into a camera. So we have a pattern here that is somewhat complex. And we can do better than this, though. So these animals are crepuscular, they feed in the evening, and then they're active early morning as well. Remember those resistance parameters? Well, we can use the resistance parameter here as well. So if we have this temporal encounter rate change, so this pattern we estimate, we can simultaneously estimate resistance. And in this case, resistance is the density of paths or riverbeds, which is what they move along. And here's an example of an animal who lives there at the black dot, and this is its home range. And the home range is longer to the right because there's less resistance to movement to the right. What we can do is, in a single model, put this temporal effect together with this resistance effect, and I'll show you the little animation. So the animation is going to start at time 0 and run through, and then it'll cycle. So here we go, starting at time 0. It decreases, little pause, then dips down, middle of day, it's not active at all. It just reverts to its activity center, sits there, then in the evening it goes up to its biggest range, down a bit, little blip in early morning, then down again midday to being completely inactive. So from this, we've managed to estimate not only how it uses its habitat on average over time, but how it uses its habitat within day and when it's most active and most inactive within day, at the cost of 4 extra parameters here. This, though, is its average use. So at any particular time, the animal is only going to be in one point. It's not going to be in all these points in which it could possibly be. This is just an average across many, many days where it would likely encounter rate at various points in space at various times. So in order to model the actual movement of individuals, we'd really like to integrate into that a movement model of the individual and then be able to predict where the individual is going to be, given where it was last seen and when it was last seen. And I'm going to do that in the next example, but not quite that. So the next example is camera trap data in the Garaholi Reserve. And the survey region is the green bit here, and the cameras are the black crosses. And this is data first analyzed and reported by Dorazio and Currens in the paper cited at the bottom here. And what we're going to be interested in is modeling how the animal's distribution in space, so where it's expected to be encountered, changes given where it was last encountered. So something akin to a movement model. It's an empirical model, though it doesn't have any sort of mechanistic steps of the animal moving. It just gives an average movement, as you'll see in a moment. So these are the traps again, the cameras. I've just squashed it into a square area for convenience. The circles are the cameras, and I'm just going to focus on this sub-region here and one animal. So here's the sub-region and the animal. When the lines hit, the gray and the red are the same animal, and when the lines hit a trap, that means it was detected at that trap. I've separated it into the first half of the survey and the second half. So the first half of the survey I've joined with gray lines, and the second half I've joined with red lines. And the reason I did that was to make it apparent that where this animal distributes itself in space depends on where it was last. So it tended to spend time in the southern part of its home range at the beginning of the survey. And although its activity center was fixed, let's say, and we presume, it explored the southern part first, and then in this particular survey, it then explored the northern part. And so therefore, if we know where it was last detected, we can get a better prediction of where it's going to be than just somewhere in its activity center. Detected in second half, it's more likely going to be in the north. So we then built a model that involves two extra parameters that I'm going to illustrate again with an animation. So this picture here is now this picture, except of the white dots are the camera traps again. The activity center, which is a red cross on the left, is this tiny little dot on the right. Excuse me. And the thing I've superimposed on this is a sort of a color scheme which shows expected encounter rates, a number of encounters, which is highest at the activity center, and then the animal tends not to move very far away, and it's symmetric in this relatively simple example. And what's going to happen is this animal is going to move around its activity center in a way that we can't see or know, and then it'll be detected somewhere, and I'll put a little star like that when it's detected. So when it's detected somewhere, we now know that the animal is more likely to be next detected in the vicinity of its last detection, except it's not likely to be detected symmetrically about this vicinity because it's always attracted to its activity center. So it's going to be more likely that the red bit shifts here, but it's not going to shift symmetrically about this point. It's always going to be pulled in to the activity center. And so you'll see that going on when I do the animation. The animation looks a little bit like, I think, London being bombed in the Blitz, but it's not bombing. It's the activity center, or the expected number of encounters moving around. So I'll just run the animation for you to see what I mean. This work is in progress work. We submitted a paper on it a while back, so it's not finished yet. All right, animation. Detection. You can see it's being pulled towards the detection every time. Pull towards detection, pull towards detection. So this is taking account of the spatio-temporal correlation in its movement about its activity center, which is getting some way towards a movement model, because we can now predict that it will at various times will be in certain vicinities more likely to be there than in other vicinities. It's an empirical model, though. We don't have any sort of mechanistic model for how it moves about space built into that. And so that was something we were interested in because we were interested in modeling how it moved about space, just using camera tap times of detections. But it turned out that when we did that, the abundance estimate changed a whole lot. So let's just look at the left picture, which is the estimated population size. And this is Bayesian inference, so these are posteriors. So when we build that movement in, that's the red curve, we get a posterior abundance around 350, whereas when we don't build the movement in, we assume independent detection everywhere at any time, we get a abundance estimate of only 200. Now, this remains to be verified because this is a really big effect, but it does seem that we need to model the spatio-temporal correlation in order to get reliable estimates of density. So aside from being of interest itself, how they move around the activity centers, it actually may influence estimates of density. Okay, that was a whistle-stop tour through some of the examples where I've tried to illustrate how we get from really very little data, just photographs on cameras or hair snares and possibly times attached. We try to build up a picture of how the population behaves and moves. So we're nowhere near the case that we were with Trump supporters storming the Capitol, where we could pretty much follow the whole population all the time, know exactly where everyone was, what roads they used, etc. But we've got a reasonable way towards that, and there's a lot of prospect of getting even further towards that when we are able to combine GPS tag data with the camera trap data. Now that has been done, but not been done in a very elegant way so far. So I'm sure that in the next year or possibly even less, people will integrate these two kinds of data into a way that lets us make even more specific and accurate inferences about habitat preferences, movement, conductivity, and so on. And that really is all I wanted to say, except I'm going to take an unashamed plug for my institution and an MSc that we started running. So 2 years ago, we started running an MSc in statistical ecology at my university. And if you're interested, you should contact me or look on the webpage. I'll remove the plug and finish the talk. Thank you very much.
Thank you so much. That was really great to see. We have a few questions for you. Samira would like to know, and she apologizes in advance if it's a naive question, but is your definition of resistance similar to what is traditionally defined as matrix resistance when animals cross different habitats?
I'm not sure, because I'm not entirely sure what matrix resistance means. That resistance idea, as far as I can tell, came from geneticists who had defined resistance of populations moving over much, much longer time periods. They'd come up with exactly the same idea. So I suspect it's a very similar idea.
Oh, okay. And the next one she has is, how would we have to change these dynamics when we study animals like crickets or birds that move over much smaller areas?
Crickets, don't know. That sounds fairly challenging. These big carnivores are really easy in a way. I mean, big carnivores are not easy in that your sample sizes are small. Whereas with crickets and birds, you get probably much higher sample sizes. But bird movement on the sort of real-time movement is really difficult to deal with unless you can actually observe in real time where the bird is, just because they tend to move a lot faster than tigers or leopards or anything. But in principle, the same ideas should be applicable. It's just whether you can gather the data. So that's not a very satisfactory response, I'm afraid. But in principle, the ideas should apply. Crickets may be difficult because everything I've spoken about here, I've assumed you can identify the individuals.
Mm-hmm.
Now, there's quite a lot of work on what if you can't identify them all, or what if you can't identify any of them. And there's a lot of work on methods developing with that, but the quality of the data degrades. You get less information if you can't identify the individuals, and with less information, you're less able to make such powerful ecological inferences.
Right. I actually had a question. Sorry, there's another question from a participant, which I'll cover first. Divya would like to know, how do we apply some of these concepts to species for whom individual identification is difficult? Building occupancy random encounter models with relevant covariates is part of it, perhaps, but are there any other ideas or developments?
Yeah, individual, if you don't get individual ID, everything becomes a lot more difficult. So there are methods there now. If you get some individual IDs, and so that may be some animals are individually identifiable within the species, or it may be you can mark some. So there's a whole literature in spatial capture recapture called mark recite, where you've marked a subset, and out of that subset, you get the information on how much they move. And then assuming that's a random sample of the population as a whole, you can then use the number of encounters from the unmarked individuals. Combining those two, you can get similar sorts of inference. There are also methods so other than in-mixture or occupancy models, there are methods where spatial capture recapture is being adapted for the case where you can't identify any individuals. So you'd mentioned occupancy, you didn't mention in-mixture. So the in-mixture is a way where you get more information than occupancy, because with in-mixture you get counts rather than presence-absence at points. And you can make similar kinds of inferences, but the sort of bottom line is, if you lose the individual identities, you lose quite a lot of information, and in particular about how they move. Right. Because an encounter here and an encounter here, you have no idea if those are the same animal or not. And as a consequence, you just aren't able to make as strong or as many inferences about movement as you could with marked individuals.
Right. I had a question that was similar to Samira's, but looking at the marine ecosystem or looking at movements of marine megafauna, how applicable would this be in that case, where the home ranges, well, the home ranges are smaller at their particular, let's say, breeding site or foraging site, but the migrations are vast. And I'm sorry if this is a very naive question.
No, that's I think that's a very good question. So in principle, it's all these ideas are all applicable to exactly that scenario. So if you only surveyed in one part of their migration area, so I don't know whether the breeding area maybe, then you can only draw inferences about that, and you can apply exactly these kinds of methods, providing you can identify the individuals and where they were detected. And in principle, you can then just build a model which is multi-state. So it has a model for what goes on in their breeding area, and then they go to a feeding area maybe. And so you have a model sort of like the birth-death model had. You have a transition from here to here. So instead of transitioning from unborn to born, you transition from this location to that location. So that adds a transition parameter. And then within each of these areas, you can model their movement, providing you get the information within each area.
Right. All right. We don't have any more questions, but I'm sure participants will have questions after the talk. And, David, you can answer them on Slack afterwards. But thank you so much for joining us today, considering the time difference and everything. And it was really interesting to know about some of these models, because personally, I am not so well aware, and definitely gives you something to think about and perhaps even apply in future. But thank you again, David, and we hope you'll be able to join us again at SCCS Bangalore in person soon.
Thank you very much, and thanks for inviting me. It was a real pleasure being here, and I'll look on the Slack and see if I can answer questions if any appear.
Perfect. Thank you so much. And thank you everyone for joining us from your homes or wherever you are. We'll see you tomorrow for day 3